Hello everyone, and very welcome to the last session of the 31st PAGE meeting here in A Coruña. And while you are gathering here, I can tell you I am Siv Jönsson, and I am a member of the Scientific Organizing Committee of PAGE, and soon to become a dinosaur. And I'm also working at Uppsala University and Prometheus. So, before we start this session, I would like you to listen very carefully now, because after the closing remarks, there will be your opportunity to give feedback on what you think the PAGE meeting, how it should be, and any input you have. And then we gather here in the corner over there, and Thomas Dorlo will be there and lead the discussion. But please feel welcome to come. It's very valuable for us to hear what you think we should do for next year. Okay, with that, it's a pleasure to go back to the session now. And this will be a session about model diagnostics, and we have three very interesting talks. And first, I would like to welcome my colleague, Dr. Martin Bergstrand. Martin is a principal consultant and an MIDD platform scientific lead at Prometheus. And the talk he will give is named Application Relevant Shrinkage Measures. Shoot.
Yes, thank you, Steve, for that introduction. And nice that so many is coming to watch this last session. So the objective of this presentation is to illustrate how relevant shrinkage metrics can be derived for secondary parameters, such as, for example, AUC and Cmax, and how that shrinkage may affect sequential PKPD analysis. So shrinkage in the form of eta and epsilon shrinkage for nonlinear mixed effects modeling was first introduced in connection to a PAGE in 2007 by Rada Savik and Mats Karlsson. And since then, publications on shrinkage has primarily focused on the impact of shrinkage on model diagnostics and strategies to overcome that. Recently, there is also a proposed method to overcome this issue of eta shrinkage when it comes to TDM applications. So the figure up here showing is a snippet from the original 2007 PAGE presentation that gives the definitions of eta and epsilon shrinkage and a graphical illustration of how these two measures depend on the information content of the data. So from now on, when I'm talking about shrinkage, I will be focusing on eta shrinkage and the similar type of shrinkage for other parameters. So before we get into the actual talk, I want to touch a bit about some common misconceptions regarding eta shrinkage. So first of all, we need to keep in mind that the traditional definition of eta shrinkage and the typical way that it's reported is in fact a mean shrinkage for all individuals in an analysis data set. And that shrinkage for a specific individual depends on the design of the data, the applied model, but also the underlying true model parameters. And under normal circumstances, the individual shrinkage is difficult to assess. However, differences in shrinkage between different subgroups, such as those with difference in study design and or baseline characteristics, can easily be assessed. So here in the figure, we see an example of the shrunk post-hoc random effects distribution for analysis data set compared to the true omega distribution. And this is for all individuals, but if we start breaking that up into different subgroups, we can see that the overall shrinkage of 45% actually is made up by one subgroup with a semi-rich sampling design, both with oral and IV dosing, which has a eta shrinkage for this random effect of 17%, and then other subgroups ranging all the way up to 71% shrinkage for a subgroup with the most sparse design. So in the literature, you often come across a statement similar to this one, where it says, in general, shrinkage indicates that the model is over-parameterized for the data that is available. The first recommendation is to simplify the model. And I disagree with this and similar statements. High shrinkage in itself is not a reason to simplify a model at the cost of a worse model fit. And we can illustrate why this is from the example that we see in the figures. Here we have two random effects for clearance and oral bioavailability F, where the shrinkage is 16 and 45% respectively. However, if you derive the secondary parameter of main interest of clearance over F based on these two parameters, the shrinkage for that parameter is only 7%. And if we go one step further to the AUC at steady state that depends on this clearance over F parameter, we will see that the shrinkage further diminishes all the way to 4%. And simplifying this model by removing the random effect on, for example, the oral bioavailability would in no way have improved this model or led to a better, for instance, sequential PK/PD analysis. So, alluding to that, high shrinkage, eta shrinkage has been suggested to introduce bias for sequential PK/PD analysis. And while this can be true under some circumstances, this presentation aims to demonstrate that eta shrinkage per se is not a good metric to evaluate this. So first, how do we derive shrinkage for secondary parameters? Shrinkage can be derived for a secondary model parameter that depends on at least one or more random effects by defining it as in percent 100 times 1 minus the standard deviation for the log of the secondary parameter based on the post-hoc parameters divided by the standard deviation for the log of simulated secondary parameters based on the same applied model. And examples of secondary parameters for which we can derive this type of shrinkage is PK, secondary PK parameters, exposure metrics, but also different type of PD metrics such as nadir or steady state response. And these secondary parameters in turn can be used for subgroup comparison and or sequential analysis such as exposure response assessment. So to look into this, we have applied a simulation re-estimation approach. We applied a one-compartment PK model with first-order absorption and with fixed allometric scaling for disposition parameters according to the table. We simulated an analysis data set with 4 different subgroups, 100 subjects in each. Subgroup A having a semi-rich sampling design and both oral and IV dosing. Group B having semi-rich sampling but only oral dosing. And subgroup C and D having sparse and very sparse design in the form of 2 or 1 trough PK sample. We also simulated a uniform weight range for this population. And we have hypothesized 3 different continuous drug response variables with underlying linear exposure-response relationships. Response variable 1 being related to AUC at steady state, response variable 2 being related to Cmax at steady state, and response variable 3 being related to Cmax for the first dose. Then we applied 3 different scenarios. Scenario 1 with 5 different dose levels ranging from 100 to 800 milligram. Scenario 2 with only a single dose level for all individuals. And scenario 3 also with a single dose level, but in addition we removed the weight covariate when re-estimating model parameters. So this gave rise to a very busy table, but don't worry, I'll take you through it. Some of the highlights. So as expected, eta shrinkage increased with a more sparse sampling design for all random effects. Parameters clearance and volume was less shrunk than the corresponding etas due to the variability explained by the weight covariate in scenario 1 and 2. Clearance over F and volume over F was less shrunk than the underlying parameters clearance, volume, and bioavailability F, due to having a high degree of identifiability. And for scenario 1, the AUC at steady state was less shrunk than clearance over F, due to part of the variability being explained by differences in dose, whereas for scenario 2 and 3, where we had a single dose level, the shrinkage in AUC steady state and the shrinkage for clearance over F was identical. But most noticeably, even when eta shrinkage is very high, the shrinkage in exposure metrics can be relatively low. And this can be exemplified for the subgroup C with only 2 trough samples, and where in all cases, for all scenarios, shrinkage in these exposure metrics is relatively small. So what happens then to the sequential PK/PD analysis for these examples? So scenario 1 with a wide dose range can be summarized easily by saying that no meaningful bias in exposure response parameters was seen for any of the subgroups under in this scenario. For scenario 2, we had a similar result in terms of that there was no meaningful bias in exposure response parameters for any of the subgroups and/or response variables. However, it's quite noticeable that with more and more sparse design, we get a lower R-squared value and consequently a higher uncertainty in the estimated exposure response parameters. And this is really due to the inert correlation between individual shrinkage and individual exposure metrics uncertainty, their relative standard errors, for example. If we go to the last scenario, where we had 1 dose level and no weight covariate, we had no meaningful bias in exposure response parameters for subgroups A, B, and C, and not either for subgroup D, the very sparse design, when we considered the exposure metrics AUC and Cmax at steady state. However, for subgroup D, when we were looking at the response variable 3 that was related to Cmax at after a single dose, we had a very high shrinkage, you can see here, of 75%, and as a consequence, we had a strong positive bias in the estimated slope, and consequently a negative bias for the intercept. And this is interesting to see that when we introduce large shrinkage, we do not get a diminishing exposure-response relationship, but an apparent more steep exposure-response relationship. And this can easily be understood by this very theoretical example, where we have, again, considering a linear exposure-response relationship, and as we introduce a perfectly symmetrical shrinkage for all the exposure metrics of 20, 40, 60, and 80%, we see how the consequent slope, when you relate the response to this exposure, gets steeper and steeper. But not only that, we actually also distort the shape of the relationship and introduce more of a power function type of relationship. And if we look at this instead for an Emax type exposure-response relationship, here exemplified with 3 different Emax models with varying EC50 values, we can see that when we introduce shrinkage in a similar way, we again introduce an apparent more steep exposure relationships in the form of a higher hill than true hill coefficient, so a positive bias for the hill coefficient. Depending on where the true underlying EC50 is in relationship to the bulk of the exposure metrics, we may also introduce bias for the estimated EC50, in the way that when the true EC50 is greater than the observed exposures, we introduce a negative bias for the estimated EC50, whereas when the true EC50 is lower than the observed exposures, we introduce a positive bias for EC50. But in conclusion, the most important factor here is that eta shrinkage is not a good metric to judge the validity of a sequential PK/PD analysis approach. The shrinkage for the exposure parameter of interest is a better metric to consider. And even with very sparse PK sampling, the shrinkage in exposure parameters can often be sufficiently low to allow for an unbiased sequential PK/PD analysis approach. And when shrinkage for the exposure parameter of interest is high, for example, greater than 25%, exposure response parameter estimates estimated with a sequential PK/PD analysis approach can be substantially biased. And simulation re-estimation approaches can be used to evaluate if a specific study design will allow for an unbiased sequential PK/PD analysis, and it can allow us the possibility to design more informative studies and devise efficient and accurate analysis approaches. With that, I would like to conclude with acknowledge and thank Professor Joel Tarning and Dr. Richard Höglund and the whole Clinical Pharmacology Department at Model Oxford Tropical Medicine Research Unit for hosting me during my recent sabbatical and for valuable input on this. And last but not least, all of my colleagues at Prometheus for continuous inspiration and for valuable input. And I want to thank you all for listening.
So thank you very much, Martin, for this very timely presentation. And now it's open for questions. And remember to present yourself and your affiliation.
Yes. Hi, Sven Mensing, EPI. I was wondering for the PK parameters, so very nice presentation, by the way. Do you need a full covariance matrix for the omega, or can this also be done with a diagonal matrix only?
It's not dependent on that. You would want to have a full omega matrix if you have substantial correlations between the random effects, of course. For this example, I did not use a full omega matrix for the simulated example.
Hi, Jonathan French, Metrum Research Group. Martin, excellent talk. Thank you so much for all the sort of insights in the different ways, some of the misconceptions. I think those are really important to highlight. I noticed the definition of your definition of shrinkage for the secondary parameters is on the standard deviation of the parameter itself, whereas when we're talking about sort of the shrinkage for ETAs, we're talking essentially about a residual. So the shrinkage here for the secondary parameters, it could include variability due to doses and things like that. So can you speak to sort of maybe how that could be potentially impacting sort of our interpretation of that measure of shrinkage?
Definitely. No, that's at the heart of this presentation because, as I said, the title was Application-Relevant Shrinkage Metrics. And if we want to have a shrinkage metric to judge the validity for a sequential PK/PD analysis approach or for subgroup analysis between two groups, this might differ. But if we want to do a sequential PK/PD analysis approach, I think it is the shrinkage that includes the variability coming from fixed effects, such as dose, covariance, and so on, that is important. Whereas, of course, we know that if we would instead want to maybe look at the subgroup differences where you want to remove the variability that is explained by maybe imbalance in doses between groups, you would want to derive a metric that does not include that source of variability. But that's user can easily define this. Yeah.
Okay, I don't see any further questions, so thank you again, Martin. Yes. Next, we have pharmacist Sarah Baklouti, who is a PhD student in Toulouse, at the University of Toulouse and at the University Hospital in Toulouse. And Sarah will now present on multivariate exact discrepancy, a new method for PK/PD model evaluation. Please.
Thank you. So, good morning, everybody. First of all, I want to thank the organizers who gave me the opportunity to present our work. So I am going to talk about our new method, multivariate exact discrepancy, that allows to evaluate a model. Thank you. So in hospital, we perform a therapeutic drug monitoring to ensure the efficacy of a drug and to limit its toxicity. And usually, we measure one concentration in a patient, and we want to determine the exposure. So we have to use a population pharmacokinetic model. We use POP-PK model published, built by other teams and available on databases such as PubMed. and we have to select a compatible model with the patient's concentration. So to do that, we can use graphical tools such as visual predictive check or VPC. In this example, the drug was administered by IV route with a bolus, and the kinetic is monocompartmental. So here are the VPC provided by the first model, and you can see that the patient's concentration falls within the VPC interval, so the model seems compatible. Although the VPC provided by the second model are really different from the first, the patient's concentration falls again within the VPC interval, so the model seems compatible again. The third model could be compatible, but we don't know if it's really the case. And finally, the fourth model is incompatible. So in this example, we can see that the VPC don't discriminate between the first and the second models. In some cases, we measure two concentrations in one patient, and we consider that if both concentrations fall within the VPC interval, so the model is compatible. But if you look at the second example on the right, you can see that both concentrations fall within the VPC interval, but this situation is pharmacologically impossible after an IV administration, as the second concentration is higher than the first concentration. So the VPC don't allow to select a compatible model to perform a therapeutic drug monitoring. That's why we choose to develop another method based on simulation. So to use our method, you have to perform simulations with the tested model, and you have to simulate individuals with the same sampling time, the same dosage regimen, and the same covariance values as the patient. And we choose to represent the concentration simulated at the first sampling time on the x-axis, and the concentration simulated at the second sampling time on the y-axis. And here are the first simulated individuals, and here I have added the second simulated individuals, and here is the cloud of points I obtained when I simulate 1 million of individuals. But this cloud of points isn't really informative about the distribution of the concentrations, and that's why we choose to to represent the density of the cloud of points and the density curve. So you can see here that as we go to the middle of the cloud of points, the density increases. And now, if I add my patient points, so in the middle of both distribution of simulated concentration, you can see that my patient's concentration is outside of the cloud of points. If we compute the multivariate exact discrepancy for this patient, so we have to evaluate the percentage of simulated kinetics with a lower density value. So in this example, the med value for our patients is below 1 divided by 1 million. So finally, even for this example, the VPC don't allow to detect a compatible model for my patients. Then, if we summarize the property of med, so we have the method provides 1 med per kinetic profile. If we have a low med, then we have a low probability to observe the patient concentrations, but if we have a high med, we have a high probability to observe the patient's concentration. So, in other words, the med can be interpreted as a p-value. I have introduced the concept with only one individual, but you can compute the med for several individuals, and all the med are independent between individuals. Now, if you want to evaluate the model, you can use the method. So you have to compute the med for each individual. And if the model fits the data, we expect that the simulated concentration distribution is similar to the observed concentration distribution. So in this example, you can see that the observations represented in red here are inside of the cloud of points. So if we represent the med distribution, you can see that the med distribution follows a uniform distribution on 0 to 1. On the contrary, in this second example, you can see that there are several points outside of the cloud of points. So we have several lower values. And if we look at the histogram of the med, you can see that it doesn't follow a uniform distribution on 0 to 1. We have evaluated the performance of med with a preliminary simulation example. So in the literature, there are several models built with a monocompartmental model, but we know that the majority of drugs follow at least a bicompartmental kinetics. So we think that the tools available today don't allow to detect this misspecification in the structural model. So we wanted to evaluate the med in this context. We simulated observations with a bicompartmental model, and we have simulated 200 sets of 200 individuals with 2 concentrations per kinetic profile. The first concentrations were simulated during the first stage of the kinetic, and the second concentrations were simulated during the second stage of the kinetic. And we have simulated 200 sets with a very highly bicompartmental model and with a less and less bicompartmental model until we get a monocompartmental model. Then we built a monocompartmental model that best fits the data, and we evaluate the percentage of sets identified as coming from a bicompartmental model. We choose to compare the med to the PDE. So the PDE isn't designed to evaluate a whole kinetic profile, but it seems to be the closest graphical tool available today. I have represented here on the y-axis the percentage of sets identified as coming from a bicompartmental model, and on the x-axis the different bicompartmental kinetic from the most bicompartmental here to the least, so the monocompartmental kinetics. And here I have added the result provided by the med, and you can see that the med allowed to detect that the sets came from bicompartmental models. You can also see that 5% of observations coming from a monocompartmental kinetics are identified as coming from bicompartmental models. And this result is consistent with the significance level of our test. Here I have added the results provided by the PDE, and you can see that the PDE allowed to detect that the observations are simulated by bicompartmental models only for kinetics very highly bicompartmental. So in this example, the med seems more powerful than the PDE to detect a misspecification in the structural model. So to conclude, we propose an evaluation method usable for one individual in the context of therapeutic drug monitoring and for several individuals in the model building process. The MED provides one p-value, the method provides one MED per kinetic profile, and I have presented the concept with only 2 concentrations per kinetic profiles, but you can compute the MED with several observations, with more observations per individual. The MED are independent between individuals and fully exploit the correlations between observations made in one individual. Moreover, we know the exact distribution of MED, and it is expected to follow a uniform distribution on 0 to 1 when the kinetics come from the model. And finally, this method is really easy to implement, but larger scale performance validation is now required. Thank you for listening, and I wait for your questions.
Thank you very much, Sarah. Very interesting, and I think we have questions.
Yes, Leonid Gibiansky, Quanfarm. I'm wondering, looking on your idea of detecting like p-value for the first problem, alternative, you can simulate number of individuals with the same sampling time, feed it back to the model, and look on the distribution of the objective function and consider where your objective function, individual objective function for that subject sit in that distribution. I think this p-values that you get this way and that way should be closely related. So it would be interesting to check.
Yes, I didn't test to compute the likelihood function, but thank you for the idea, and I will test it when I got back to Toulouse.
Leiden University. I thought it was a really nice presentation, very clear to follow, and I really liked how you compared this distribution to these different data points. And I was wondering, because you say if it's a uniform distribution, then you would say it's a good model, basically. Do you then also evaluate what is a good uniform distribution?
I use a statistical test. That's your questions?
Oh yeah, did you use a metric, or you just use it, you look at the visual representation of the distribution?
No, not. We perform a statistical test, a Kolmogorov-Smirnov test, to evaluate the uniform distribution of the MED, but you can use other tests if you want.
Okay, thank you very much.
Hello, this is Gang from AstraZeneca. So I was wondering if we consider a real clinical trial situation that samples are often taken at different time points, meaning we have different patients, the sampling is like random. Would we see such a pattern when you plot this plot, right? Because of the samplings are random, so the difference between the two concentrations would be, I'm not sure, yeah. So have you tested this scenario?
You want to know how we simulate the concentrations in the simulation example? That's right?
Yeah, I mean, just if you take samples at different time points, yes. not regular points.
Yes, because we built the monocompartmental models with the simulated concentrations. And we want, it's not possible to build good model with only concentrations sampled at the same times. So it's to evaluate the performance of the method in this context.
Okay, thank you.
Hi, Jonathan French, Metrum Research Group. Thanks for a really nice talk. Can you give a little bit more detail about how you did the density estimation, and whether that was from like you were assuming a normal Gaussian, or you're doing some non-parametric smoothing for non-parametric density estimation?
How I estimate the density? I use all these simulations are made on R, and I use a package available on R. It's a kernel smoothing package.
Okay. And so as you moved from 2 dimensions to maybe higher dimensional data, do you have a sense for how well that density estimation is going to work?
I'm sorry, can you repeat the questions?
You were showing it for 2-dimensional, 2 data points.
Yes.
So as you move to higher dimensions, maybe you're doing for 5 data points. Now your density estimation is in 5-dimensional space.
Yes. With the KS package, you can compute the density with more observations per individual. So I remember until 6 concentrations per individual. And now I haven't found another package to evaluate the density with more observations per individuals, but if someone will work about it.
Okay, thanks.
Okay, thank you. I actually have also a question. So I always want to understand how you do this. So do you have a tool ready to derive this med?
I don't. I didn't have a tool. We use her, so with the KS package. Okay. So it's just 4 lines, 4 code lines. So.
Okay. All right. I think there is no. There is yet another question, and we have time for it. So please.
Your approach through therapeutic drug monitoring. Now, if you have two patients, and for one you identify that a given model is consistent with the data, and for another you identify that it's not, would you then use different models for different patients?
Yes. Yes, of course. Thank you for the question. At the hospital, we got several models, and we can choose between different models to select the most appropriate model for the patients. And this method allows to make a choice between the all possibility.
And may I have just a quick other question? So could you comment on how you do the simulation? So you use established models, include population analysis that has been reported. So then you sample from the estimated distributions, or
yes.
Okay.
I use a PK model available on PubMed, and I code this model on R. And if I think the model is consistent, and after I perform simulations by random sampling.
Yeah. So then what you showed is basically 2-dimensional credibility intervals, or not intervals, regions.
Sorry?
Then it's basically 2-dimensional credibility regions that you computed. But we can follow up on this.
In this example, it's 2-dimensional, but you can apply the method with more dimensional. But
I'm not questioning, but it's related to the Bayesian approach of computing credibility intervals or credibility regions in 2 dimension.
Yeah.
Yeah. Okay. We can follow.
Okay.
Thank you very much, Sarah. Okay, and now it's time for the last presentation of this page meeting. It is a bit sad, but then we are very happy that it will be a presentation by Dr. Stephanie Hennig. Stephanie is a pharmacist, and she's also a very experienced researcher that is now working as a senior pharmacometrics consultant at Certara. And Stephanie will present the project on behalf of a larger team at Certara. So please, Stephanie.
Thank you very much for the nice introduction, Eve. I also have an unusual disclaimer to make right at the start. I know we're in the Model Diagnostics session here, as Eve said before, but I'm also part of the organizing committee, and we couldn't come up with a better session name in the end. And we knew that this talk is probably not quite a diagnostic talk, so no diagnostic tips necessarily for me. And as Steve said, I am presenting that on behalf of a larger team at Cetara, and I want to highlight especially Anna and Wayne, who've done a lot of the hands-on work, and also Keshup, who's planted the seed probably about a year ago now on this project, so I should acknowledge them. And I'm sure some of the background slides here are no news to this audience. We all know that exposure response information is at the heart of determining the safety and effectiveness of a drug during drug development. ER evaluates the risk-benefit ratio for dose selection, justification, and confirmation. And ER analysis has become an integral part of clinical drug development and regulatory decision-making. However, we've noticed that there's been a change in the last probably year or two, and that the guidelines are somewhat still lagging behind. So we all know we use a lot of different approaches when we talk about exposure response analysis. There are a lot of different methods to use that contribute to the collective information of this. And a lot of that is dependent, of course, on the type of the response variable. There is logistic regression, for example, that we use for binary endpoints. There's time to event analysis that we use for time-varying endpoints, and there's longitudinal analysis that we use for progressive endpoints. And there's probably a lot more that we can do and that we're all doing. For the interest of this talk, I will focus basically on logistic regression analysis and looking at binary endpoints. So logistic regression analysis, like I said, is used for binary response variables. These are types of data where we know the patient had an event or not an event as a side effect, or these are responders or non-responder subjects. And while the logistic regression can handle multiple exploratory variables of the probability of an event, we focus again in our talk here on only exposure driving this probability. So again, I won't probably not going into detail of the equations, that should be all very familiar. I'd like to highlight just the figure and a few things in that figure in more detail because of showing that a few more times throughout my talk. So we have here the probability of an event, and down at the bottom of the x-axis we have time average concentration. The dots at the top here are the time average concentration values for all subjects that had an event. And the ones at the bottom here, in this case, they're not quite at zero. They should be at zero, but you'll see in the animation later why they're not. These are the time average concentrations for subjects that didn't have an event. The red curve here is, in this case, representing the logistic regression model fit for the data that we are showing with the dots. So, of course, when you're doing an exposure response analysis, you're not looking at just one single exposure matrix, and you might select the particular exposure matrix that you're interested in very carefully as well. Often you look at multiple exposure matrices at the same time, and you might base that, hopefully, on pharmacological plausibility. You will also base your choice of exposure matrix on prior analysis findings or on the timing of your event that you have. So exposure matrix that you might look at is minimum concentrations, maximum concentrations, AUC at cycle 1 after the first dose or at steady state, etc. On this slide, I want to highlight in particular the difference between time average concentration and average concentration at steady state. And maybe, again, that is not something that's new to this particular audience, but I think it is getting mixed up quite often and commonly. So for clarification, I wanted to highlight that. So we start with average concentration at steady state. that is derived from the AUC at steady state. And the AUC at steady state is the nominal dose that is given to a patient times the bioavailability, of course, divided by clearance. And then the average concentration at steady state is derived from that AUC at steady state divided by the dosing interval. In comparison, the time average concentration is derived by the cumulative AUC, which is the cumulative exposure since the start of treatment for a patient, divided by a time of interest. So in case of having an event, it's quite easy. Our time of interest is the time of the event. And the benefits of the time average concentration is, of course, that it can very nicely look at each individual patient's dose interruptions, dose modifications, reductions, and so on. But the problem with the time averaged concentration is that you don't have a time for subjects that are censored in the study. So these censored subjects are subjects that have either dropped out, have not been followed up, or have not had an event throughout the treatment period or trial period. So our question was basically to evaluate the impact of deriving time average concentration for subjects without an event and what kind of impact that has on our exposure-response relationships. So throughout the talk, we have used different ways of deriving time average concentrations for censored subjects. As I said below, the time for subjects with an event here is very clear. You have that, you know that data. But for all subjects without an event, we used either the time of the end of treatment, or we added 7 days, 14 days, 21 days, or 28 days to that time. And these are basically chosen based on either washout periods or an alternative time that's commonly shown is the data log base time, because that's when no more data is collected. So what does this impact have just maybe now diagnostic-wise on our figure here? Again, maybe ignore the red curve. We're not changing this for the moment. We're keeping that the same to illustrate the model fit for the red dots in this figure. So this is now we're using now for the censored subjects down at the bottom here, the time averaged concentration when we use the time of the end of treatment period. Now what happens to this when we add 7 days, you have the purple dots. If we add 14 days, we have the gray dots, green dots with 21 days and 28 days. So one very obvious thing that you see that the concentrations for the subjects with an event don't change because they still have the event time at the same time, and we only did the imputation for the subjects down the bottom here. But here you see a shift to the left. So with that shift of the left, you will have some implications on your model fit afterwards. And I will show you these now. So we've used for our little simulation a data set that we had from a real life data set, but we had to modify that for sensitivity and ethical reasons. So we created 3 virtual populations with a sample size of 50, 100, and 200 subjects. We all dosed the patients the same, 60 milligram once daily for 4 cycles, 28 days. The exposure in our case was the maximum concentration that was driving the effects based on a 1-compartment model with first-order elimination. The events were simulated based on a proportional odds model with Markov components. You see the structure of our model up here, and all subjects that ended up in our grade 0 basket here were the subjects which didn't have an event, and all subjects that ended up in the grade 1 or grade 2 compartment, they were the subjects grouped in the yes, they had an event and they were in a so-called any event type group, and we used the first time, at the time of the first event they had. We also changed the ER relationship by the strength of the ER relationships from 0.05 to one-fold of the original ER relationship that we used by wearing the Emacs parameters, and all simulations and logistic regressions after that were performed in within RStudio. I don't know where my RStudio went, but it's gone from the slide. So just to show you the simulated data sets that we got from this model, illustrated here by the event rate. And we see that we saw an increase in the event rate with increasing drug effect. We saw similar event rates across the 3 different study sizes, so just what we expected and what we wanted to see. The other thing we looked at, of course, was the distribution of the time of the first events for our subjects with an event, and they all had their event at steady state or after steady state, and the distribution was that the events occurred any time from cycle 1 to cycle 4. Now here are the results of our logistic regressions. I will illustrate the results on one example. This is the 200 patient data set, and we use here the 0.5-fold effect relationship, just because it's the prettiest to show in the figures, really. And we've got here our typical logistic regression analysis figures, and you see them, the fit is the blue line here with the confidence intervals. We have our dots of the concentrations for the events and the non-event subjects up the top. We also show the p-value up the top here, and this first plot is for when we use the end of treatment time for the censored subjects. I'm going to go slowly and a few times backwards and forwards, hopefully, through these animations, and to show you. So one of the things that I want you to look at is the first quarter here, the green and the red group, and the shape of the slope of the model fit. So now we're adding 7 days to the end of treatment time. You see a bit of a change. We're adding 14 days, we're adding 21 days, and we're adding 28 days. And I can go back on that again. So there was no relationship here, and we're going to a highly significant relationship there. The only thing we've changed is the time we use to derive the C-average concentrations for censored subjects in this. So here are the p-values in the table, and basically we saw that the p-values on the slope parameter decreased with an increasing time used to derive time average concentrations for censored subjects. Now you want to know how does it look like for all the other simulations and logistic regressions that we did afterwards. We saw exactly the same trend for all ER relationships across all the tested data sets, irrespective of sample size. Of course, we saw that with increasing sample size, significant p-values were reached at lower imputed time values than at other times. Then the next step that we did was look at comparison to our steady average concentration at steady state. And we noticed with interest here that when the ER relationship is strong, so that's at the, this is at the bottom of each table. This is a very slow pointer, sorry. So you see here that when we have the effect, drug effect here, gets stronger towards the bottom of each of these tables. We see that they're all in agreement, there are significant relationships. However, when we have weaker drug effects and weaker relationships, our C-average, time average concentration relationships do not show the same relationships as the steady state concentration. Next one, sorry. We've also evaluated looking at different clearances, how the PK affects, and we're working on more simulations in regards to that. So there's going to be more information coming in the future, hopefully, on that one. However, again, we noticed the same trends here, that p-values on the slope parameters decreased with increasing time values to derive time-averaged concentration for censored subjects, irrespective of the change in clearance that we made. And again, we saw that the relationships don't necessarily align. In conclusion, the time used for censored subjects to derive time average concentration can have a significant impact on logistic regression ER relationships. And as a note here, we've seen similar things with Cox models as well and others. So we would highly caution choosing the time for the censored subjects. Of course, if you can use time to event analysis with time varying exposures, that's probably the best way and the highly suggested, but that's not always possible. So the things that we found most helpful in all our work is to explore in our analysis the exposures across our subjects, look very carefully at the exposures of subjects with an event and no events, do a sensitivity analysis when you pick a specific time that you want to use for the censored subjects, and investigate always multiple exposures, and most importantly, consider your pharmacological plausibility of choosing a particular exposure matrix, because it all comes back, it has impact on subsequent event projection, dose selection, and go, no-go decisions. So thank you very much for your attention. I'm taking questions.
So thank you very much, Stephanie, and I see there are questions. Please.
Yes, I think the problem is much, much more difficult than you describe.
Yeah.
It's not only the time of event for sensory subject, it's a time of event for any subject, because if you probably consider it simple case with no significant accumulation and constant scheduler, but nowadays you always have titration, you have accumulation, you have cases when you have weekly doses, first 12 weeks, and then bi-weekly, tri-weekly doses. So time to event exposure is bogus measure. It's always biased. It's always impose exposure-response relationship. And it looks good, but actually it's always biased.
Yeah.
So we need to be very, very careful when considering measure for exposure-response analysis. In fact, with the current dosing schemes, there are no good exposure measures at all. It's much more complex. Your presentation kind of focus on one small aspect of this.
Yes.
But I just want to kind of spread the knowledge that it's not that simple.
No, thank you very much, Leo. I couldn't agree more with you, and we've looked at a lot of these things that you've just said, but I think at the end of the day, we thought a very simple example, the most simplest case to spread the message was for us the most important, because we see how it doesn't seem to be, maybe in this audience, maybe we should have gone with our talk to somewhere else, but I absolutely agree with you, yes.
Okay, I would like to give the question to the gentleman on top.
Thank you for the interesting talk, Bas Gouwloos, LPMP Consultants. So one scenario that I was maybe missing is the scenario where there is no drug effect. Because I could imagine that the fact that the way you calculate your exposure estimate is defined by the outcome leads to a risk of maybe a very high type 1 error. So did you look at that?
We only went down to 0.05 of the original. We didn't use the no drug effect, no.
Because in this scenario where, for example, you would take an unlimited time, then the exposure would be 0 for the...
Yes.
the people who didn't have the event and
Yes.
non-zero, and then it would always be significant, right?
Yes.
Thank you.
Nick Holford, University of Auckland. Stephanie, thanks for a challenging and interesting discussion, but it seems to me that logistic regression needs to go in the bin.
Maybe.
And if you have something that's related to time, then guess what? Time and an event use time to event analysis. So that seems very simple. Even the statisticians should be able to understand that. So my question to you is, you made the remark saying you should probably use time to event analysis, but sometimes you can't use it. But I don't understand when, because as you explained, you have the concentration profile, you have the event time, you know when the censored events happen, you have all the things. So why can't you do a time to event analysis?
It comes back to time, and time this time in a different aspect.
You mean work time?
Yes.
Oh, you're just lazy. Well, not you personally, but okay. It's not a valid excuse. Doing it the wrong way quickly is not an excuse for not doing it the right way, slowly. Okay.
Okay, 2 more questions, those that are in line.
Yeah, thank you. So Martin Bergström from Ethios. My comment is a bit along the same lines as some previous, but I would like to counter what Nick said in that I'm not sure that any time-to-event analysis would actually help with this. It depends on how you do that. Also, you can easily induce similar type of biases for time-to-event analysis too, depending on how you do it. But you mentioned things like dose interruptions.
Yes.
And one thing that is an important caveat is that those interruptions are often related to the response, of course.
Yes.
And here is where we really introduce a strong bias that goes in the way of hiding exposure-response relationship. Because if you are not taking your drug due to sort of a poor tolerability, you get low exposure. Yes. And it all of a sudden, those with lowest exposure are those that have most events.
Yep.
You will see reverse exposure-response relationships, which should really make you worried.
Yes, I agree with that one as well. Yes.
Yes.
Hello, Sven Mensing, AbbVie. I have a very basic question. So why did you pick the end of treatment and not the time of the last endpoint assessment?
So you can, it's a matter of terminology and wording in this. The 28 days is basically the end of either the cycle or the database lock or whatever you want to choose as that. We've seen a variety of different things that we've used. So this was just for the simulations. It was really just to illustrate, but yes.
Yep. Thank you.
Sorry, I got stressed. I forgot to say the most important thing. The one possible unbiased solution to this is Markov models using time-varying exposures, repeated training also on times when there is no events. I think that.
Thank you, Martin.
So thank you very much, Stephanie. So with this, I would like to conclude this session, and I really would like to thank the presenters, giving very timely presentations, and I also would like to thank all of you having been here listening. But now it's time for concluding the whole PAGE meeting.
Thanks, Eve. Thank you all for resisting surviving all the event till now. Before we close, after we close, don't leave, because Thomas Dorlo will take the lead to discuss potential program for next year. Your suggestions and input is really welcome. So I would like just again thank you all for being so active and contribute and enjoying to the party. Yesterday I heard it was a really good party for those who survived after 1 a.m. I'd like to give now the word to Isa Gonzalez, who is part of the local organizing committee, to make the concluding remarks. And again, applause to the organizing group.
So now we want to tell a very little story. So once upon a time, a group of pharmacometrician people decided to conquer a city in Europe, and Coruña was selected city but a goal without a plan is just a wish, so we have to have a plan. Population of pharmacometricians were distributed to Coruña, and almost 900 people arrives here. They bring computers, knowledge, and a real passion for science. So, talk about PAGE is talk about success, and this success is due to PAGE is a meeting focused in students. We have 200 posters, 240 students, and here you can, I hope that you do new relationships, more pharmacometrician friends, more collaboration, new jobs, new opportunities, and so on. The success has been possible thanks to our sponsors, so please, thanks for all of them, and our technical secretariat, AFEA and Viajes El Corte Inglés. Scientific program has been amazing, as always, and Gara dinner has been okay. PAGE, I think that you arrived to Coruña to get to conquer the heart of the city, and I hope that Coruña conquer your heart. So now it's time to back home and work very hard to prepare new spectacular works to the Bella Italia, Rome, and see you in the next meeting. I hope all of you.